Whenever you see a scientific result, whether it's in the news or in a deep dive research paper, you're really just seeing the final snapshot, the destination. But today, we're going to go behind the scenes. We're going to uncover the incredibly meticulous multi-step journey that makes sure those results are actually trustworthy in the first place. So, it all really boils down to this one simple but massive question. How do we know? Well, it's not about blind faith, that's for sure. It's all about a super rigorous, verifiable process that is designed from the ground up to produce accuracy. So let's trace that process right from the very beginning. All right, here's our roadmap. We're going to follow the complete journey of a single sample through 4 key stages. We'll start with the environment where it's all prepared, and we'll end with the systems that guarantee we can trust the final outcome. So our journey actually begins way before a single test is even run. Because think about it, to get a precise result, you first need a precise and, well, a totally controlled environment. And in any biological lab, the most fundamental part of that environment is water. I mean, it makes sense, right? Over 70% of most living things is just water. So the purity of the water you use in a lab can literally make or break an entire experiment. And it's this unique, kind of bent shape that makes water so special. It creates these positive and negative poles, basically turning every single molecule into a tiny little magnet. And that magnet is what can pull apart and dissolve all the materials we actually need to analyze. It's the ultimate solvent for life. Now, beyond the chemistry, the physical environment is governed by some super strict safety rules. And these aren't just about protecting the scientists, they are about protecting the science itself. A single contaminated glove or a misused chemical can introduce errors that corrupt a result before the real analysis even kicks off. Okay, so the stage is set. Now, the sample itself begins its critical journey. And believe it or not, this part right here is where things are most likely to go wrong. So to understand how labs really nail accuracy, we first have to understand the 3 main stages where sneaky errors can creep in. We can break it all down into 3 phases. You've got the pre-analytical phase, that's everything before the test. Then there's the analytical phase, which is during the test. And finally, the post-analytical phase, after the test is all done. It's a really simple but powerful framework for hunting down potential mistakes. Now, here's the crucial part, the thing you have to remember. Most errors happen before the sample even gets near the fancy machines. We're talking simple things like mislabeling a tube, using the wrong collection method, or storing it at the wrong temperature. Any one of those can completely invalidate a result from the very start. So once a sample arrives safely, how do we unlock its secrets? Well, this is the analytical stage, and it involves some truly fascinating techniques. I want you to think of a cell as a locked treasure chest. All the valuable information, all the good stuff is inside. So the very first step in any analysis is finding the right key or, just breaking the box open. This is basically a 2-step dance. You break, and then you sort. Over on the left, you can see all the different ways they do it. Physical, chemical, you name it, to crack those cells open. But once you have that messy mixture, you have to sort through all the pieces, which is where a real cornerstone technique, shown on the right, comes into play. And that technique is called centrifugation. Just think of it like a super-powered sorting machine. By spinning samples at incredibly high speeds, it creates this immense force that separates everything in the mixture based on things like its density and its size. But what if you want to separate multiple things from that same sample? Well, you don't just spin it once. You spin it multiple times, cranking up the speed with each round to pull out progressively smaller and lighter components. And here's what that looks like in the real world. You can see how each spin, getting faster and longer, acts like a finer and finer sieve. It systematically pulls out different parts of the cell, starting with the heavy nucleus, then the lighter mitochondria, allowing scientists to isolate exactly what they need to study. It's just a beautifully precise way to deconstruct a cell. Okay, so we've got these incredibly precise techniques, but how does that scale up? I mean, how do we trust the thousands of results a lab produces every single day? Well, for that, you need a system. The answer is a huge, comprehensive framework called laboratory quality control. It's basically the whole set of measures that are put in place to safeguard the accuracy, the reliability, and even the timeliness of every single result that leaves that lab. And this system rests on 4 key pillars that cover every single aspect of the lab's operation. It's not just about the fancy equipment. It's about the people running it, the processes they follow, and a deep commitment to learning from every single error so you can constantly improve. But get this, labs don't just get to mark their own homework. Nope. To truly earn our trust, they have to prove their competence to outside independent organizations that hold them to global standards. This validation comes in a few different forms. It can be a certification for a specific process, or a full-blown accreditation for the entire lab's competence, or even an official government license to operate. Each one is another layer of external proof that the lab really knows what it's doing. And going through this whole rigorous process to get a global stamp of approval, something like an ISO accreditation, has huge benefits. As you can see here, it's not just about bragging rights or a fancy plaque on the wall. It builds trust, it improves skills, and it opens doors all around the world. Ultimately, it all comes down to this. Achieving this level of accreditation is the ultimate seal of approval. It's proof positive that a lab's processes and its competence are top-notch and that they meet a global framework for confidence. So you've now seen this incredible system of checks, balances, and precise techniques that underpins all of scientific trust. But it leaves us with one final and really crucial question. What happens when this system fails?